Hello, everyone. My name is Jackson Wynn, part of the Forensic Science program at BCIT, and today's topic is Proficiency Testing in Forensic DNA Labs, an error rate study. Here is my overview. We're going to be going through 8 different topics, as denoted on the slide, and the first one we're going through is, of course, introduction. As we all know, DNA is so prevalent in forensics. It's one of the first things that we think about when we think of forensics. As the RCMP would say, an indispensable tool in countless criminal investigations and judicial proceedings. And even though DNA profiling is considered that indispensable tool, researchers such as Butler, Colson, and Bassett, DNA profiling is definitely prone to errors, and with common failures such as contamination and human error, which definitely can affect an examiner when they're trying to determine whether or not a DNA sample um, would um, would be identified part of the victims or the suspects or another person. Here we have our literature review. We have a forensic DNA analysis, error rates, proficiency testing, and interlaboratory control. So the first topic, forensic DNA analysis. Definitely DNA has completely changed over decades with new techniques, new methodology for examiners to use. What we see, especially with instruments, is a high sensitivity that's allowed DNA profiles to actually be analyzed only tens of a picograms. And essentially with DNA being able to be able to analyze at such a small scale definitely may result in error. Um, so what happens is, with Rivera et al., who was examining with the human belief of where forensic DNA actually lies on the accuracy scale, it actually was seen actually as the highest, with 89.95% out of all the forensic techniques. And even though that's simply the human belief of the accuracy of for forensic DNA, there definitely is that consideration of a range of error that may be prevalent in DNA. All right, and also we have our third topic, proficiency testing. Essentially, proficiency testing is essentially it's a third-party kit, usually administered by another company, which laboratories and examiners partake in. And when they have that test, they submit the results back, and then the proficiency tester can definitely examine to see where they lie on the spectrum of where with other laboratories. And essentially, what the PCAST says is the only way to establish scientifically that an examiner had been capable through appropriate empirical testing to measure how often the examiner got the correct answer. And definitely, with proficiency testing, is to understand where a laboratory needs to improve, whether they're on the right track, whether they're still doing a really great job, or they're doing a really great job. And then we have our final topic, interlaboratory quality control. And essentially, it's that comparison of laboratories. So essentially, what happened is with the quality control and comparison of between laboratories, laboratories can definitely learn from one another to improve their results. Whether like laboratory has really great technique in this section or great instrumentation, how can another laboratory definitely improve on what they have? All right, so we are going to my research question. So how has proficiency testing with collaborative testing services affected the error rate for forensic DNA profiling in 2014 to 2019 when using forensic biology, DNA mixtures, DNA semen, DNA blood, and test kits? So other research questions that are related is I wanted to understand whether the effect of aberrant allelic results and how they affect the error rate on DNA interpretation for victims and suspects. And essentially, aberrant allelic results basically means that variation in the results. So sometimes results aren't perfect. There may be some slight difference. So that's what aberrant allelic results means. And for the next question, we have what is the error rate for respective test kits between 2014 and 2018 for the interpretation for victims? And on the other hand, we want to understand the error rate for suspects for DNA interpretation. All right, so we have our hypothesis. So I wanted to understand that variation in allelic results and whether that had a significant effect on the error rate for victim DNA interpretation. And on the other hand, I want to also examine for suspect DNA interpretation. And finally, for the next hypothesis, we have each of the respective kits blood, semen mixture, forensic biology, they have no effect on the rate of the aberrant allelic results. And also want to examine if they had a positive or negative effect on the rate also of aberrant allelic results. And we're moving on to methodology. So for my methodology, there were quite a number of records on the collaborative testing services because they had so many different kits. Starting at 112 as denoted at the top. And what I wanted to do is I'd excluded topics that were irrelevant to DNA, which I found was 12. And from there, I actually determined which subject matters that did not seem prevalent or related to my topic per se. So which I turned out to be DNA interpretation, DNA parentage, and DNA database saliva, which I essentially chose the topic forensic biology, DNA blood, DNA semen, and DNA mixtures very noted at the bottom. And those were the best topics because those were the ones that seem most prevalent in a forensic laboratory. Like those are the stuff that they would get the most. And they also seem related and correlated to one another. All right, and so on a bigger format of my methodology, as we see, is from the collective subs from the collective test kits. We have our collaborative testing services that get divided into subscales, as in the middle. From the subscales, from there they get the analysis of data from using correlation and scatter plots to understand the trends and also to understand the patterns of victim DNA interpretation and suspect DNA interpretation. All right, so these are my subscales. So we have the number of aberrant allelic results, and which can get converted to the rate of percent of of aberrant allelic results. And from there, when we're understanding the reporting of DNA interpretation, we can actually definitely see the correct victim DNA identification and incorrect victim DNA identification. And also, on the other hand, we can definitely look at suspects. And from there, we convert it to like an error rate for DNA interpretations for each subject matter. All right, and we have our results. For the results, I want to understand the internal consistency. So for the one that I'm looking at is for forensic biology, and also is the same for all the other test kits, is what we notice is with the subscales, there's actually a strong internal consistency. Since the Cronbach alpha is actually greater than 0.7, these subscales are great with one another and show a strong internal consistency. And for next part, for the aberrant allelic results, for forensic biology, what you see is actually a general positive trend. There's this slope increase. What we see is the aberrant allelic results, or that variation percent, has increased over the year. And for forensic biology, it has increased at 0.2%. And also, interestingly enough, at all the other test kits for blood, semen, and mixture, there has been an increase in the aberrant allelic results across all test kits, which show that sensitivity, as talked about earlier, does affect this aberrant allelic result percentage that's found. It seems like it's on a general increase overall. Next one is for the victim and suspect DNA interpretation. For the forensic biology, as victims denoted in blue, we see that this slope decrease over time. So examiners, when interpreting for victims for the forensic biology kit, we see that their error rate for that DNA interpretation actually has decreased over the year from 2014 to 2019. And we can also say the same for suspects also, as denoted in the red. We see this decline for both of them, for suspects and victims, interestingly enough. And the great part is for the other test kits, for blood, semen, and mixtures, we actually see a decrease over time for all of them. The only ones that we note that there hasn't been a change was for on the left on the DNA blood, there hasn't been a change for the DNA interpretation for victims. While in the middle graph for DNA semen, we actually see a slight increase for suspect DNA interpretation. There's that positive increase right there. But overall, what we see is for all the test kits throughout, there has been a overall decrease in DNA error rate interpretation. So next one is I want to understand the correlation of aberrant allelic results in DNA interpretation. As noted with blood semen mixture in forensic biology for victims and suspects, what we see is with this correlation across all test kits, there is actually a lack of significant correlation between the aberrant allelic results and DNA interpretation for victims and suspects. So even though there is this increase in variation in the aberrant allelic results, it did not significantly affect at all when examiners are doing that interpretation for victims and suspects. And next part is my discussion. So what we see is we see an increase in aberrant allelic results. This may actually be due to the increase in sensitivity and instrumentation. Like I said, it's like tens of picogram, which are so immensely small. And the thing is, we shed skin cells everywhere. So it just shows that it can definitely affect the results in the end of the day, where it gets presented. But what we also see is the decrease in DNA interpretation error rate in the middle. The decrease in DNA interpretation error rate for victims and suspects has decreased. So what we see is even though the variation in allelic results have increased, DNA interpretation for victims and suspects have been on a decrease. So there's this inverse relationship almost. But the great part is also there's a lack of significant correlation between the aberrant allelic results and DNA interpretation. So even though, like I said earlier, that there's this variation in allelic results, it does not affect examiners to actually properly make that match between victims and suspects. Next one we have is our limitations. So proficiency kits have a limited scope and do not perfectly simulate a real crime. They are usually considered perfect test kits because their sample's so perfect. And in a real case, of course, DNA can be there could be traces of DNA that are degraded, and so many different factors that could potentially have on a criminal investigation and when it gets transported to an actual lab. Another thing to know is these tests are not blind. Examiners do know that they're being tested, and they're also not well controlled. So if an examiner is doing the test kit, someone definitely could come in and definitely help them, but there's no way of controlling that. And also these test kits don't provide an actual source of error where it actually the error belongs to. Does it belong to the instrumentation? Does it belong to the methodology? Does it belong to there's not enough training for the examiners? And finally, some future research for the topic, definitely blind studies to understand more about the error rate in forensic DNA analysis, just because it will show a better controlled environment in that regard. Also detailing where errors may be pertained, such as with the contamination, training, instrumentation, and also another great way is to have more proficiency tests throughout the year, just so laboratories can see a great change or growth of where they lie on the proficiency testing scale. And thank you so much. Thank you for the presentation. Thank you for listening.